hospital about 1 year ago. Before that, I was a postdoc at Duke University in the States. And before that, I did my PhD at the Technion in Israel in electrical engineering. So I'm basically expert in AI algorithms. And in the past 4 years, I'm working on different problems in medicine. Now, I am part of the iMedData AI Center. It is led by Dr. Ahuva Weiss-Meilik. So let me skip. So basically, our center is composed kind of two departments. One is a group of data engineers. The data engineers basically know to access any data in the hospital. So basically, we can access any data, do any research we want on this data. The other part is of researchers and data scientists who are actually doing the research. In addition to that, in our team, we have 2 senior nurses and a physician who is also a cloud architect. Okay, so basically we have all the range of expertise to do research in the intersection between AI and health. In general, our research center is doing many different kinds of works, collaborating both with the academia and the industry and other HMOs. The type of projects we are doing vary from just providing data for companies to doing applied research in AI. Okay, I will be focusing on the research part. So these are the different organizations that we are working with.
That's a lot of partners.
A lot of partners. The green ones are those with whom we have agreements and we actually work with them. The yellows are in the process, and the reds are an organization who we were less lucky to build something. Okay. So basically, my goal today is kind of give you a taste of what we are doing. So I have 30 minutes, right?
Around 20.
Okay. So I will present one project in slightly more details, one project in 2 minutes, and 2 projects in 10 seconds. Okay? So let's start. First one is about computational cytology. Okay, so what is a cytology? Oh, you don't see my screen, just a minute. What you see here is a scan of a glass slide in pathology. Basically, it's some kind of material taken from the human body. It can be cells, it can be tissues. Okay, it is smeared on this slide. So this is kind of an eye-bird view of the slide. And this slide is scanned with a scanner. Okay, we are getting an image, and with an image we can do image processing, and we can apply AI algorithms. Here specifically, this project is about predicting thyroid cancer. So let's take a look here. By the way, the size of this image in pixels, if I want to show the whole scan, I need a monitor like this 1,000 times, okay? So I cannot show the whole image, but I can zoom on a very, very small region of the image, okay? So basically, when a patient comes to the hospital with a suspicion of thyroid cancer, the most important test that is done is called fine needle aspiration biopsy. So that maybe I can show you in a slide. Sorry, I will come back to it in a moment, to the cats and the dogs. So basically, the biopsy is done as follows. As a fine needle is injected to the thyroid. A small amount of matter is extracted, put on the glass slide, okay? And basically, the pathologist is doing the diagnosis. The goal of the pathologist is basically to assess the risk of malignancy. If the risk is high, surgery is required. But the problem is that about 30% of the cases are indeterminate. Okay, it has a very nice name. The physician called it suspicious or abnormal and so on. But basically, it means that the pathologist doesn't know if it's cancer or not. Okay, about 50% of these cases eventually go to thyroid gland is removed, and then another biopsy is done, which is 100% accurate. Okay, the problem is that 65% of post-surgery biopsies are found benign. It means that there are many surgeries that are unnecessary, and our goal is to build an algorithm to try to say if surgery is required or not. Okay? So how we do it with AI? For that, I need to go back to these images of cats and dogs and kind of give a 1-minute brief introduction to machine learning or AI. So probably the most common example of AI or example task is doing classification of images. So for example, here, the task is to classify between dogs and cats. How we do it? We basically show to the algorithm or the neural network or any other machine learning model, we need to show many examples. What do I mean by example? An example is basically the data, which in this example is a dog, and the label that says that in this image you have the dog. Okay? The training process is basically very, very similar to the way we teach children about the world. Okay? So, for example, if I say to my kid that, listen, this is a dog, so he knows that the exact type of this dog is really a dog. The question is what happens when I show him to my kid a different type of dog. And then the question if my kid, or in this case the algorithm, knows to differentiate between them. So this is in a nutshell the idea. Once we finish the training, we are doing a test. The same as we are testing kids in the school, we are testing the algorithm. And the way we do it, we show to the algorithm a new image that was never seen before. He needs to do the prediction, and then we need to have the ground truth to evaluate if the prediction was correct or not. So what we are doing in the case of the thyroid cancer, we basically take these images, okay? And the ground truth is what happened after the surgery. I don't want to predict what the physician will say about this image, because I know that in many cases the physician doesn't know. I want to teach the algorithm something better, which is what happened to this patient after the surgery. So basically, this is what we've done. This project started 4 years ago at Duke University, where I was a postdoc. We collected the biggest data set in the field of thyroid cancer, which is basically all cases from 2006. Overall, we have a data set of more than 100 terabytes. Okay? And sorry?
That's a lot of bytes.
That's a lot of bytes.
Yeah.
So it's like 5,000. The storage I have in this laptop, 5,000 times. That was a mistake to lift.
You insulted your computer.
Okay. So actually, we weren't the first one to solve a similar problem in the field of pathology. There are actually many startup companies that are working in this field. And one related project that I was working on was in predicting prostate cancer. Now, what's the difference here? In prostate, what we have here is a whole tissue. So basically, we have a lot of data, a lot of information on the slide. In contrast, in thyroid cytopathology, what is interesting are only these tiny cells. All the rest are blood cells, and they are irrelevant. So the problem here is basically that the signal-to-noise ratio is very, very low. Okay? So we tried an algorithm that was recently published that performs almost perfect on prostate cancer, and in our case it just failed. So from almost, so I'm talking here with the metric I used here is the area under the curve. It's the most common in this field. The higher is the better. So 1 is perfect score. Here we got 96 for prostate. But basically, the algorithm failed in our case. And the question was why. So one problem we got is the signal-to-noise ratio, as I said. The other thing is that we actually don't know if we have the signal inside. Because as I told before, in 30% of the cases, the physicians do not know. It means that we even don't know if these cells can tell us the true story. And we actually don't know it until this point. Eventually, the algorithm that we designed was basically mimicking a pathologist. What the pathologist is doing is first finding the relevant cells, okay, ignoring all the blood cells, and then doing the classification. So just like a taste of the results we got. Again, the higher in these plots is the better. This is the performance of our algorithm, and we compared it to 3 expert cytopathologists from Duke doing the same task, assessing the same slides exactly. And what we can see is that our performance are comparable to the best of the cytopathologists. After we finished this project, we started working. The next phase of this project was something more ambitious. So basically, until this point, we used a scanner that costs about $200,000. And there was an NIH grant that we applied to and received that where the goal was to promote healthcare in low and middle income countries. So basically, the idea of using AI to save manpower and to help pathologists is very appealing in low and middle income countries. For example, we are working with a center in Tanzania, in Africa. In whole Tanzania, there are about 6 pathologists in the whole country, okay? But the problem with applying our method there is that we cannot use this scanner because they just don't have these scanners. And then, therefore, we asked ourselves if we can replace this with a $200 smartphone that we connected to a microscope with an adapter that costs like $15 that we bought from Amazon. So to do that, we needed to basically answer 3 questions. One is whether the quality of the image is taken by a smartphone is good enough. And actually, you see that it is. So this is an example from the scanner, and this one is from the smartphone. And actually, what you see is that the image from the smartphone is more in focus. Is more in focus. Is even of better quality. And I can explain later why.
Wow.
So one challenge kind of was solved. The second question was what will be the effect on the AI algorithm that was trained on the scanner images when we showed or when we are using camera images. And actually, if I show these 2 images to anyone, and for sure for pathologists, they will see that they are basically the same, right? The question is whether the AI algorithm sees these 2 images as the same and provides the same prediction or classification. So I will answer this in a moment. The last question is whether we can get enough image area when taking the camera, the images with the camera. So I didn't tell you before, but to do the classification with images from the scanner, we use basically 500 images per scan. And clearly, it is not feasible to ask someone in Tanzania to take 500 images for each prediction. So basically, I depict it like looking with a straw onto the slide. Okay. So I'll show what happened when we applied first the algorithm to the images from the camera. Remember, exactly the same content, 2 different acquisition devices. What you see here is the degradation in the results from the scanner to the mobile images. Okay, we lost about 15%. Okay, and the only reason is the limitations of AI. This is the only reason, because we should expect from good algorithm not to do mistakes like this one. Okay. I will finish this part of the talk by just giving one sentence of how we solved it. Basically, we used a technique called data augmentation. And we did it on the scanner images that were used to train. Okay, so we didn't change anything about the camera images when testing. We just applied different kinds of manipulations on the images of the scanner when we trained the algorithm. And the idea was to reduce the effect of the color on the classification. And the results we got was basically that we closed the gap. So you can see here in the method that we propose, okay, that the predictions based on the camera and the scanner were almost the same. Okay, so because I have only 1 minute, I will move to the. I will use them to describe 3 projects, just so you can see the range of the different projects that we have in our center. The second one is predicting patient deterioration in internal departments. Okay, so it sounds completely different project. What we want to do here, we want to alarm the staff, the medical staff, that a patient is going to deteriorate in the future. Okay, the solution with AI exactly the same. We take a neural network or a different model. We show the data that we currently have about the patient, mainly electronic medical records. Okay, the ground truth, what we teach the algorithm to predict, is what happened to the patient in the future. Okay, so again, almost the same machine learning techniques are used for this task. And just to show the result, so here I'm comparing the green line is called early warning score. It is the score currently used at the hospital. And our method wins this score by more than 200%. Okay? And some other projects that we have, we know to predict length of the surgeries. We are now working on management of queues. We are working on predicting readmissions to the hospital. And there is a big project of basically taking a natural text that describes the history of the patient and transforming it into a data that is basically associated with a timeline. So basically we can kind of summarize the story of the patient over a timeline. That's it. Questions?
Yeah. Great. The results, are they public? Are they open? Do you share them with the world? Are they cooperation projects? How does that work?
So the work on pathology, we have several publications. It eventually became a multi-center study with many collaborators, including from University of Toronto, Duke, UNC, and others. The other projects, part of them are we already published, and part of them we will publish in the future.
One thing is publishing, and the other one is the algorithms and the data sets, and how open are you going to be with that?
We are not. The goal eventually is to commercialize this. Okay.
So you work. Is it going to be a spin out, or are you already working?
It's early to say. The next phase is going to be to do a prospective experiment. So actually, we know that we can alarm the physicians on time, but we don't know the effect of it. We don't know that it will save lives.
Because we have another 2 speakers.
One more question.
One more question, please.
I have a question about the variables you use in the models. For instance, if you look at the length of surgery, and you take some of the variables and put them in the model, then how accurate will the model be as you are looking at the same time at variables you can use for improve the operation and length of surgery. So for the accuracy of the model, can you tell me more about it? How do you?
Yeah. So basically, and this is kind of the difference between the projects where we do where we process images, and the other projects where we do processing on EMR data. When you do in AI methods like deep learning, which are the most common in image analysis, basically the variables are the image, okay? And this basically means that the algorithm is kind of a black box, and we cannot really explain it. It's very hard.
Yeah, it's stable.
Yeah. On the other hand, in the projects where we work on EMR data, which is a tabular data, because it's much smaller data, we use other algorithms that they are much more interpretable. So we basically know which of the variables is more important. So for example here, okay, where we predict patient deterioration, we know that the most important parameter is the age of the patient, which is not surprising. Okay, but in general we can assess the effect of each variable, and this way we also know if the prediction makes sense. So for example, we see that to predict patient deterioration, other important variables are different lab tests and vital signs like heart rate, temperature, and blood pressure.
Yeah, but for instance, in the group of patients all aged 80 or above, age doesn't contribute that much anymore to our model. So the models. Wat jij doet op het moment dat je aan bepaalde factoren sleutelt, gaan die modellen moeten zo precies voorspellen.
Oké.
Dus je weet wat je hebt, ga je dit kunnen doen. Maar dat is je model niet meer voorspelbaar.
Okay, I want to thank you very much for having this presentation, and we want you to remind you of the Netherlands. We have Dutch stroopwafels, which is very sweet. You've been to Amsterdam,
Awesome.
I think you liked it. If you come again to Amsterdam, we'd love to have lunch with you and get to know your brain a lot more, because it was fascinating. Thank you again very much.
Thanks. Can I have a picture? Can I have a picture?